Voilà. Okay, so let's start again. So I have great pleasure to have here today Manolis Kellis, who is really a young, let's say, young rising star. He is now he's the professor in computer science at the Massachusetts Institute of Technology, and especially in the area of computational biology. He's a member also of the Broad Institute at MIT, at Harvard, and he heads the computational biology group at MIT as principal investigator in computer science and artificial intelligence at MIT. So his contributions essentially are related to genomics, human genetics, epigenomics, gene regulation, and genome evolution. He leads a number of projects such as the NIH Roadmap Epigenomics project, which he had led, and also the ENCODE, GenCode, and mod-encode projects. So he's especially interested now in human disease, to relate all this on human disease. For instance, such as the diabetes, Alzheimer disease, schizophrenia, obesity, and cancer. So he is of Greek origin. So he moved with his family, in fact, to France from Greece. And then he came to the US in 1993. He was working, he was doing his PhD with Eric Lander at MIT. And then afterwards, he won especially an award for his thesis, the best thesis in computer science. And he is now, he then afterwards became the professor at MIT, as I said to you already. So his track record is very impressive. So he is, I think, if I calculate this well, he has the age of 44, but he has a age index already at Google Scholar of 122 and 114,000 citations around already. So I would say at the age of 60 or 65, it will be enormous and skyrocketing. In fact, if he has already at that age, in fact, the citation index as high. So his work is on comparative genomics. And at the beginning, he was working on the yeast genome. He published a number of papers. He developed the computational approaches and methods, and which has been, in fact, afterwards also applied in other species. He has done also a lot of work on epigenomics, and he is working on several disease states, such as Alzheimer disease, obesity, and he's also in the consortium, is a member of the ge-netic consortium, the genotype tissue expression. Now he's, in fact, his work also he's doing some interesting work that is, let's say, interesting to me personally. He has done also work on the blood-brain barrier. You have to see his recent papers on the blood-brain barrier, which are really, really interesting. And in fact, so he's also active on the web. You know, he has a number of YouTube videos you can watch. And I came across Manolis because, so there's a podcast which is called the Friedman podcast. And Friedman is interviewing a lot of very high-level scientists. So I came across this, and then I heard that, in fact, he's very influenced by French culture. He knows very well French songs. He likes, for instance, Gainsbourg, I think is one of his, let's say, I mean, he likes this a lot. And he can even cites songs from French French songs, and that's extremely nice to have someone with such a broad culture. I think he speaks also a lot of languages, including French, Greek, of course. I think you speak also German, right?
Ein bisschen. Ich habe Deutsch studiert 5 Jahre lang.
Ja, Sie sprechen ein bisschen Deutsch. It's very good. So we can even talk, I mean, also German together.
Puedo también hablar español un poquito. Lo estudio también y lo hablo.
Okay. Great. So I'm very pleased to have you here today. So it's the first time that, in fact, we talk together. You will be in August, as you told me, in France, and then we'll have a, let's say, chat again and a good dinner with good wine. And at the end, of course, I mean, all our prestigious speakers, they will receive wine bottles because we are here in Bordeaux. So we will send you two, let's say, high-level, let's say, I mean, pretty outstanding wine bottles to your home. Okay, so the floor is yours now. Thank you very much.
It's a real pleasure to be here. Bonjour à tous. Et je suis très content de prendre des questions en français aussi, donc ne soyez pas timide. Je peux répondre dans les deux langues si nécessaire. So what I'll tell you today about is our work on trying to translate genetic findings into therapeutics. And specifically, how do we understand and dissect disease circuitry at single cell resolution? And how do we use this dissection to then guide manipulation of that disease circuitry to actually reverse disease phenotypes? The basis of our work starts with genetic variation. Why? Because genetic variation gives us causality, because it tells us that these genetic variants that are associated with a phenotype like body mass index and obesity are in fact playing a causal role. These are inherited variants, and that tells us that there's something in these loci that is clearly leading to disease. And that allows us to completely de novo discover new disease mechanisms, understand new target genes, understand new therapeutics, and enable precision medicine and personalized medicine. The challenge, however, is that if you look within these regions of genetic association, in 93% of cases, there are simply no protein altering variants. All of the genetic variants are simply non-coding. In this particular case, for the FTO locus, the fat and obesity associated locus, which is the strongest genetic association with obesity, you see that there are 89 common variants that are co-inherited together, none of which is in fact hitting an exon. They're all sitting in the first intron of the FTO gene and a small part of the second intron. And the exons are these tiny little dots here, which basically tells us that we don't actually know the target gene. Many people have assumed that the target gene would be the FTO gene itself, where the locus is encoded. But what we showed in our own work is that the true target genes are actually sitting 1.2 million nucleotides away, several genes over, and 600,000 nucleotides away. So not knowing that there's a protein coding variant means that there might actually be very, very long range linking, and that we might not even know the target gene. We also don't know the causal variant because there are multiple variants that are co-inherited, any one of which could actually be the causal variant. We don't know the cell type of action, we don't know the relevant pathways, and we don't know the mechanisms. So that's what our work is trying to address. How do we do that? We basically integrate this genetic data that includes both common and rare variants with massive profiling of the transcriptome and the epigenome across many, many different tissues from healthy individuals, but also across many different individuals for the same tissue and cell type across both healthy and disease samples to understand how genetic variants contribute to this difference in molecular phenotypes in DNA accessibility, histone modification, DNA methylation, that together make up the three signatures of the epigenome, but also in the transcription of those cells, and how non-genetic variants also affect this from the environment and from other sources of variation, including behavior, nutrition, exposures, and so on and so forth. We then integrate all these data sets in computational methods to predict driver genes, regions, and cell types using Bayesian statistics, using deep learning, using causality analysis, networks, and so on and so forth. That leads us to a series of predictions about what are the drivers, which we then go and validate in extremely high throughput cell culture assays, many of which our lab has developed, and also in moderate high throughput mouse models using multiplexing and also traditional single candidate approaches. And of course, we disseminate the results and continue the cycle all over again. What does that give us? That gives us a new insight into these regions of association. Instead of saying, oh, there's something going on in this region of this AFF1 gene that is associated with disease, we can say, here are the specific genetic variants. Here are the specific cell types where these regions are active. Here are the specific gene regulatory region in each of the cell types. And here are the upstream regulators that control these regions. And then lastly, here are the downstream target genes that are responsible for the action of this region in human disease. And you can see here that one of the candidate targets is AFF1, but there are two additional targets that are several genes over that are predicted to be linked with this region of association for cholesterol. When we apply these methodologies to the first slide that I showed you on the FTO locus that is associated with obesity, we're finding that the circuitry in itself does not implicate the FTO gene. Instead, the two genes that are implicated are IRX3 and IRX5. They're sitting respectively 1.2 million nucleotides away from the causal variant and 600,000 nucleotides away from the causal variant after several intermediate genes. The analysis that we're doing at the motif level and using comparative genomics and evolutionary conservation allows us to now pinpoint the predicted causal variant as a T to C alteration, which changes this AT-rich motif, which is normally bound by this AT-rich interacting domain protein, ARID5B, that binds this motif in a large super enhancer, which is 12,000 nucleotides long, one of the largest enhancers in the human genome. And then by binding there, this regulator represses IRX3 and IRX5. That basically means that these genes have now an altered function. And what we showed in collaboration with Melina Klausnitzer and a number of other collaborators is that IRX3 and IRX5 are newly discovered master regulators of a process known as thermogenesis or heat generation. That process acts inside our fat cells and can basically make a shift from storing energy into burning energy. So all of the excess calories that you have in your diet can be stored as white fat, or they can be burned as heat in a process known as browning of these white adipocytes or beiging, whereby they accumulate a large number of mitochondria. And these are the energy producing cells, factories inside our cells. And these mitochondria give the name of brown or beige adipocytes. And they are depolarizing the mitochondrial membrane, causing the loss of energy rather than the storage of energy in ATP and ultimately in lipids. So when the motif for this repressor is disrupted, the repressor no longer binds, these two genes are overexpressed, and they repress thermogenesis, leading instead to lipogenesis and ultimately obesity. Why are we excited about this circuitry? First of all, because we now understand the mechanism of disease, and we find that the FTO locus itself has nothing to do with the FTO gene. That's the first reason. We now understand the circuitry. But the second reason is that understanding the circuitry means that we can now start manipulating the circuitry. That means that we can now start altering the disease phenotypes. How? We can, number one, decrease or increase the expression of the downstream target genes to see how we can shift back and forth between lean and obese phenotypes. Number two, we can increase or decrease the expression of ARID5B. Number three, we can actually directly manipulate these locus using CRISPR-Cas9 genome editing with C2T or T2C single nucleotide edits. That allows us to now shift back and forth between these lean and obese phenotypes. And in every single case, we found that everything works like a switch. You can basically flip it back and forth between the two. Even when we take primary adipocytes from CC homozygous risk individuals, where that motif is simply abolished, by editing the C into a T, we're able to now shift to lean phenotypes. And we're finding that thermogenesis, measured as the relative oxygen consumption rate, both uncoupled and in isoproternal stimulation, we find that this basal rate of oxygen consumption rate, which was very low for the risk individuals, is in fact not only much higher for the edited allele by changing only 1 out of 3 billion nucleotides in the cells of these individuals, but we can also show that in the stimulated conditions, thermogenesis simply turns on completely. So that means that we can restore the cellular phenotypes by perturbing this pathway. But we can go a step further and look at the organismal phenotypes. And what we see is that when we put a repression on mice that are normal, using a dominant negative construct driven by an adipose promoter for IRX3, one of the 2 target genes, we find that in fact, these mice not only start out leaner, but are unable to gain weight. You see that the control mice gain weight in a high-fat diet versus a normal diet, but these dominant negative IRX3 mice are in fact unable to gain weight. Instead, what you see is that their fat stores are completely depleted without changing their exercise, without changing their diet, without changing anything about their behavior. Instead, what they're doing is that they're burning fat when they're awake and when they're sleeping. So that's what we want to do, but we have 120,000 locations in the genome where we want to do this. So how do we do this systematically? Well, we build reference epigenomes to predict the disease-relevant tissues, regulators, and cell types. We then combine genetic, epigenetic, and transcriptional variation in the context of disease and across individuals to understand how genetic effects manifest at the epigenomic and at the transcriptional level. We then apply these methods at single cell resolution for both epigenomic and transcriptional variation. We then study cell type specific genetic effects by integrating single cell data and bulk data with genetic association studies with disease and genetic association studies with these intermediate molecular phenotypes. We then go beyond a single phenotype by looking at multi-phenotype integration using medical records, imputation, and also expression and genetics. We then look across many tissues together to look at the convergence of these pathways across tissues and across cell types, and also to recognize biomarkers of these changes that are in accessible organs in obesity, in cancer, in Alzheimer's, and overall aging. And then lastly, we use high-throughput methods to dissect the circuitry of these locations. So let's dive right in. As part of the Epigenomic Roadmap project and the ENCODE project and multiple other large-scale consortia, we've basically been working with teams of international investigators to profile a number of epigenomic, DNA accessibility, DNA methylation, and gene expression data sets in both embryonic and adult tissues, in multiple regions of the brain, as well as induced pluripotent cells and embryonic stem cells, and in vitro differentiated cells, as well as primary cells from these individuals. We profile promoter, enhancer, transcription, polycomb, heterochromatin, and activity-associated histone modification marks, and integrate them together using computational methods for learning the underlying hidden state of the chromatin based on the observations of these vectors of individual marks. This allows us to now annotate these different classes of regulatory elements across hundreds of different cell types. What we're learning from this is that promoters, for example, are extremely cell type invariant, whereas enhancers are extremely cell type specific. You can also see that some genes are off in nearly all of the cell types, but they turn on, as you can see here in the promoter and the transcribed region, from the right to the left for this PAX5 gene. They can turn on in very specific contexts. The other thing we're learning is how to interpret non-coding variation. If we see genetic variants that are associated with disease within this locus, we can then ask what are the tissues and cell types where these genetic intergenic locations that are presumably gene regulatory, what cell types do they turn on? But even more importantly, we can ask what other genes turn on at the same time when these gene regulatory regions turn on. And instead of saying that potentially genetic variants here will target any of these nearby genes, they might in fact target the PAX5 gene itself, which is coordinately active with these gene regulatory regions. So we can use coordinated activity as a signature for linking together non-coding regions to their target genes. And we can use the regulatory motif enrichments of these genetic loci across the entire genome to start predicting the upstream regulators responsible for these, the control of these regions. So how do we find relevant cell types and tissues for every one of these genetic traits? What we can do is ask for every one of these traits, what are the genetic locations, the loci, the regions of association, which I'm showing here in these large boxes. And within these regions, what are the specific SNPs, the single nucleotide polymorphisms, the variants that are associated with each of these traits? And what's really exciting in being a genomics lab is that we can basically do this systematically across every single trait in parallel. And then we can use these different traits as controls for each other to understand how are genetic variants associated with height or diabetes or blood pressure or cholesterol different from each other in their epigenomic signatures. And in particular, we are finding that if you look at the enhancers that are turning on in stem cells, they overlap specifically genetic variants associated with height. If you look at genetic variants associated with type 1 diabetes instead, they localize specifically in the enhancers of immune cells. Blood pressure variants localizing enhancers active in heart. Cholesterol variants localizing enhancers active in liver, which basically starts giving us a little bit of a diagonal here that says that different traits likely act in different cell types and tissues because these enrichments are very specific, using these traits as controls against each other. And this is real data. This is how clean these images look. If you look at height, you see very specific enrichment for embryonic stem cells and really not much more. If you look at immune traits, you see very specific enrichments in T cells and B cells. If you look at blood pressure, there's only a single enrichment here in the left ventricle of the heart. That means that enhancers that act in the left ventricle contain a large number of these genetic variants that are associated with blood pressure. If you look at fasting glucose-related traits, you see the strongest enrichment is specifically in pancreatic islets, which makes sense given the biology of type 2 diabetes. If you look at cholesterol, very strong enrichments for liver. And if you start looking at inflammatory bowel disease, you see a double signature of both immune and inflammatory cells, as well as digestive tissues, as you would expect for inflammatory bowel disease, suggesting that both tissues are in fact contributing to the genetic epidemiology and the causal roots of IBD. If you look at Alzheimer's disease, one would naively expect that the brain would be the one lighting up, because obviously Alzheimer's is affecting cognition and is leading to neurodegeneration. But what we found, surprisingly, was that genetic variants associated with Alzheimer's were instead localizing specifically in enhancers of CD14 plus immune cells. These are the signatures of the circulating blood monocytes and also the tissue-resident macrophages, including the microglia, which are the resident immune cells of the brain. So that basically tells us that perhaps the genetic risk for Alzheimer's does not localize in neurons or astrocytes or oligodendrocytes that make up the majority of cells inside our brain, but instead localize specifically in the immune cells of the brain. To validate these findings, we basically sorted neurons, microglia, and oligodendrocytes separately, and then carried out enhancer profiling with chromatin immunoprecipitation for H3, histone H3, lysine 27 acetylation. And what we found is that indeed the enrichment for genetic variants through linkage disequilibrium score regression, LDSC, the enrichment is specifically pointing to microglia as the causal cell type underlying Alzheimer's disease, and certainly not neurons or oligodendrocytes. So we published a paper back in 2015 suggesting that these epigenomic signals in human, but also in mouse, reveal this immune basis for Alzheimer's disease by showing that when mice that are a model of Alzheimer's, the CKP25 mouse, are in fact undergoing neurodegeneration, very early before the cognitive symptoms appear, you see already dysregulation of the immune pathways, but only later in time do you see dysregulation of the neuronal pathways, indicating that perhaps inflammation and immune cell function is the causal component of Alzheimer's disease. Since then, we have expanded our methods from 127 epigenomes in 2015 to 834 epigenomes in a paper that was submitted in 2019, but was just published a few weeks ago, where we basically go from 54 enriched traits to 534 enriched traits, which are giving us annotations for 30,000 different variants. This is 1/4 of the 120,000 loci that are associated with complex traits in human disease. These are highly specific associations, and these give us very precise biological hypotheses on the mechanistic basis for hundreds of thousands of these loci. So if you start looking at the same association, but now in a graph form, you see that genetic variants associated with schizophrenia, depression, neuroticism, but also with intelligence, educational attainment, and cognitive performance are localizing in enhancers active in brain. Instead, Alzheimer's localizes in enhancers active in immune cells. And you can see this complex network with hubs of very tissue-specific connections, but also many traits that are sitting here in the middle in these highways between these hubs, where you see multiple traits being, multiple tissues being implicated in the same trait. And you can see here a pie chart showing the diversity of different tissues that are implicated in each trait. We can take one example of coronary artery disease, which was one of the most pleiotropic traits, one of the most complex in terms of the diversity of the underlying tissue enrichment. And we can now start asking about partitioning that complexity into different components. For example, you see that QT interval is localizing only in heart, educational attainment only in brain, Alzheimer's, both brain and immune cells, body mass index, multiple internal organs, and health span, you see this diversity of tissues. With coronary artery disease, you see liver, coronary artery itself, thyroid, adipose, and a diversity of other tissues. But what's really phenomenal and super interesting is that when we take all of the genetic variants associated with coronary artery disease and we partition them into subsets that are active in liver versus coronary artery, etc., we're finding that the corresponding pathways are dramatically different. So we can start dissecting the complexity of CAD into different subparts. Associated with liver, you see functions with, lipid transport and lipid metabolism and LDL, etc. Associated with coronary artery, instead, you see epithelial and blood coagulation and so on and so forth. And what's really interesting as well is that the comorbidity patterns and the co-association patterns of coronary artery are themselves getting partitioned by this exercise with different traits associated with subsets of variants with within different epigenomic annotations. We can go a step further and start dissecting that complexity at the level of individual genetic loci. And in particular, if you look at the same example of coronary artery disease and all of these same tissues, you can go down locus after locus and ask, is this locus active in liver or in, say, coronary artery? And you can see that the loci themselves get partitioned. And we can start finding the specific examples of cell types, target genes, causal variants, and upstream regulators for each of these intermediate molecular phenotypes. So we can basically find these genetic variants associated with this locus here are in fact very strongly pointing to PCSK9, one of the big success stories of genomics, which is now a very successful drug against coronary artery disease and other related pathways. If you look at this second locus here, you see that this localizes very specifically in coronary artery. And indeed, the target gene appears to be linked to these genetic variants. You see two different loci, both of which are linked to the same gene, which is active in coronary artery. And you see other loci, for example, this one here, that show both signatures of liver and coronary artery together, linking multiple tissues to multiple genes and converging across different pathways to the same ultimate disease. So we basically made these predictions for 120,000 loci, and we sorry, for 30,000 loci, and we have made them all available, in this locus here, in this link here, where we can start dissecting both the circuitry, the activity and modules of these regions, and also the GWAS SNP localization and fine mapping across a quarter of the 120,000 loci associated with disease. But all of this work is assuming that epigenomic variation is only profiled in reference epigenomes, and we don't have access to the dramatic variation that happens in the epigenome of different disease individuals and different healthy individuals. So what we looked at next is how genetic variation correlates with epigenomic variation and transcriptional variation between individuals and also in the context of disease. Our goal is to bridge the gap between genetic variation and disease by looking at the path of causality through intermediate tissues, regulatory regions, genes, and endophenotypes on the way to disease. The challenge, however, is that these nice unidirectional arrows that we have are not expanding beyond the genetic variation. Namely, if you see a correlation between enhanced reactivity and disease, or the expression of a particular gene and disease, this could be reverse causality. This could be that the disease itself is causing the gene expression change rather than the other way around. Or it could be that the gene is increasing in expression not to cause disease, but to combat disease. And it could also be that both are changing due to some environmental change that is affecting both this gene and the disease independently of each other. So we need to develop new computational methods to understand the causality to get these unidirectional arrows back. To do this, we basically teamed up with David Bennett and Phil de Jager to look at a cohort of individuals that were profiled longitudinally in their old age until they died and they donated their brain. And after the fact, we can see how many people developed Alzheimer's disease and how many people did not. That's the phenotype. We can then ask, how is the epigenome of these individuals and the transcriptome of these individuals changing? And how is that related to genetic variation? So the first thing we did is look at how genetic variation at conception can predict the epigenome and the transcriptome of these post-mortem samples at 93 years of age. The second thing we did is ask how related is this epigenomic and transcriptional variation to phenotypic variation. And of course, the third thing is putting them all together to understand causality. So let's dive right in the correlation between genetic variation and epigenomics. What we're finding is 50,000 genetic loci across the genome that are dramatically significantly associated with disease, with methylation. So here on the x-axis, I'm showing you genetic variation, and on the y-axis, I'm showing you epigenomic variation. And this is after covariate correction. That's why the points are diffuse across both axes. But what we're seeing very clearly is that with a p-value of 10 to the minus 205, halfway down our plot here, you can very strongly predict the epigenome of a person based on their genetics at birth. This is very powerful because the genetic variants are co-inherited at birth. They are not a consequence of the methylation change. And that allows us to now start predicting methylation. So we can use genetics to methylation from methylation quantitative trait locus or QTL studies, the same way that we can look at genetics to disease across many more individuals. But because this is a simpler phenotype, it's more oligogenic, we can in fact use many fewer individuals than what we need for bridging that very large gap from genetics all the way to disease. So that's the first success. The second one is that we can actually directly correlate the methylation variation with disease. But the downside there is that this is a bidirectional arrow. The methylation changes could be a consequence of the disease rather than the other way around. But we can combine all of these analyses together and predict or impute methylation directly from the genetics across 74,000 people. Because here we only need a blood draw to get the genetics of a person. We don't need their brain. So people are much more generous with their blood than with their brain. You can then predict the methylation in the brain for these individuals from their genetics and then start relating these imputed methylation with disease. Why is that powerful? Because it's just a genetic component of that methylation. It's not the full methylation, some of which is environmental. It's only the genetic methylation, and therefore we get a unidirectional arrow if that genetic variation correlates with disease. So we can actually get at causality and a lot more power because we have many, many more individuals. So that allows us to now discover new loci that are associated with disease and multiple contributing SNPs that together are predictive of the methylation status of every one of these regulatory regions across the genome. We can use that to start discovering new loci and within these loci to understand what are the driver genes. We can use these across multiple intermediate phenotypes from genetics to methylation to transcription to disease. And we can start predicting both in GWAS loci that are shown in purple here and in subthreshold loci that are shown in gray, the mediating genes shown here in colors, and which ones of them are risk-inducing in red or protective in green, in blue, based on their association with the disease. And we can also ask for each of those, how much of the heritability of that locus is that one particular gene capturing, enabling us to quantify the mediation of these genetic loci to disease. But everything I've showed you so far has been at the tissue level, looking at bulk methylation in bulk tissue samples. But I showed you earlier how dramatically different are microglia versus neurons and other cell types in the context of Alzheimer's and other disorders. So what we did next is start dissecting at single cell resolution this epigenomic and transcriptional variation. So we looked at Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar, Down syndrome, autism, depression, suicide, PTSD, and aging, across 1,500 postmortem human brain samples, and across millions of cells, grouped into dozens of different cell types, and across multiple regions of the brain. And for each of those, we've profiled either the transcriptome or the epigenome, or both, to understand this incredible variability across individuals, within cell types, across disease, and across the progression of disease. In the first paper that we published nearly 2 years ago with Li Hui Tai in Nature with Mattis and Davila as the first authors, we profiled 84,000 cells across 48 individuals, half of whom had no signs of AD and no cognitive decline, And then the other half had both early and late disease symptoms with many different pathologies and dramatic loss in global cognition. We profiled to 84,000 cells, which we then classified into excitatory neurons, oligodendrocytes, oligodendrocyte progenitor cells, inhibitory neurons, endothelial cells, astrocytes, and microglia. And then for each of those cell types, we then asked what are the changes that are associated with low versus high amyloid, different Braak stages that are quantifying disease progression, different levels of cognitive decline, and the diagnosis of Alzheimer's or not, but also the sex of these individuals. And what we found is that every major cell type could be separated into both risk and non-risk associated states, which were predisposing individuals to either Alzheimer's or not. But we also found that the transcriptional differences between men and women were dramatic, and that female individuals had many more cells from the disease state than male individuals, suggesting that transcriptionally, the signature of dysregulation in Alzheimer's is much stronger in female individuals than in male individuals. We also found that the pathways associated with Alzheimer's were dramatically different between men and women. We also saw that early in the disease, we have very cell type specific changes, but late in the disease, we have much more shared changes, very often associated with damage and response to damage. We also found that men and women had dramatically different expression profiles, both before Alzheimer's, with 3,000 differentially expressed genes between men and women, but also after Alzheimer's, these differences are further increasing, suggesting that we should be profiling sex-specific epigenomes and transcriptomes, and also thinking about sex-specific and cell type-specific therapeutic targeting. In particular, we found that female individuals did not upregulate myelination pathways in oligodendrocytes, while male individuals did. That basically prompted us to then look at myelination differences, and specifically white matter loss, which we found to be significantly higher in female individuals, suggesting that myelination is now a major potential driver for Alzheimer's, suggesting that it has a lot of similarities with multiple sclerosis manifestations, but specifically in the female individuals. Since that early paper, we've basically scaled up across many more tissues and traits and cell types and individuals. We're basically totaling more than 1,500 samples, more than 7.5 million cells, and 200 billion reads, which we have now used to study many additional disorders. In the context of schizophrenia, we basically profiled 800,000 cells across 24 case and 24 control individuals. And that allows us to now go into disease loci that are associated with schizophrenia genome-wide association studies. And then for every disease locus, start predicting what are the predicted target gene that is the effector of that genetic effect for that locus, and whether it's upregulated or downregulated in schizophrenia, and what cell type in the brain are they the most up or downregulated in. Very often supported by three-dimensional chromatin conformation capture information that links together these non-coding regions to the promoters of those genes. We also found that we could classify individuals into schizophrenia or not schizophrenia based on their transcriptional signatures, and that these transcriptional signatures were global and consistent in their classification across many different cell types in the rows here. But we were surprised to find that a small number of schizophrenia individuals did not show these global transcriptional signatures in any of the cell types. And this has been reported before, that somehow mysteriously, a subset of individuals don't show transcriptional signatures. What we found instead is that those individuals show a very specific cell state, which we call schizophrenia transcriptional reversal, which might provide an alternative path to schizophrenia, or might play protective roles in transcriptional resilience for those individuals carrying that cell type, where they show the schizophrenia symptoms, but they do not show the signatures at the transcriptional level. We used our data to start predicting upstream regulators, and in particular, a module of upstream regulators that appears to have roles in early development, but also in adult changes. And we found that the genes downstream of these associations are in fact very strongly enriched for neurogenesis and multiple types of synaptic and signaling functions. In the case of ALS and FTLD, we found that the two disorders are in fact very similar to each other, and they are impacting both male and female individuals, and they're implicating a large number of pathways associated with dysregulation and autophagy and signaling, synapse function, and endocytosis. In the case of Huntington's disease, we found that the disease individuals are in fact losing the difference between these direct pathway versus indirect pathway spiny projection neurons, which are very transcriptionally distinct in control individuals, but are in fact merging together in the HD individuals. We've also expanded across brain regions from prefrontal cortex to also include mid-temporal cortex, angular gyrus, and entorhinal cortex, thalamus, hippocampus, and mammillary body. Annotating a total of 1.6 million cells across the different brain regions, each in 7 different regions, each in 48 different individuals with 24 AD and 24 controls, and then 12 male and 12 female individuals for each of those regions. That allowed us to discover new marker genes for 30 different subtypes of excitatory neurons and 23 different subtypes of inhibitory neurons that are dramatically different between these different brain regions. But it's not just excitatory and inhibitory neurons that vary dramatically between regions. If you look at astrocytes and other glial cells, you see that indeed the angular gyrus cells are very different from the mammillary body, very different from the thalamus, and so on and so forth, consistent with dramatic changes in both the function and the signatures of these different cell types in the different brain regions. And then having the multiple brain regions and having so many cells from so many individuals allows us to now quantitatively order genes and cell types according to a map of Alzheimer's progression at the single cell level. How do we do that? By building a computational function that allows us to predict the AD status of every person based on the expression patterns of every one of their cells. And we can now apply the same predictive function at the cell level to look for cells that have signatures indicative of early stages, intermediate stages, of late stages of Alzheimer's disease. And then we can see how every single gene changes along this disease time course progression. We also looked at the spatial information surrounding our single cell data. And in particular, when we focus on the hippocampus, we see that the dentate gyrus and area CA1, 2, 3, and the subiculum, and also the proximal entorhinal cortex signatures are in fact dramatically different from each other. We're finding that the clustering of these cells according to their single cell transcriptional signatures without any spatial information are in fact capturing the organization of these regions and separating granule cells from pyramidal cells and all of these different subregions completely de novo. That allows us to now start understanding the association of different subregions of the hippocampal structure with Alzheimer's disease.
I think we have a problem. He's no more connected.
No, I'm back. I just wanted to stop the vacuum from the background noise. I'm sorry. Okay, so we can now basically see that the dentate gyrus is much less predictive of Alzheimer's, and CA1 is in fact the most predictive of Alzheimer's, giving us a way to rank these different brain regions in their association. Inspired by this spatial information, we then developed methods for integrating spatial transcriptomic data with single cell data to basically look at the what I like to call the mini bulk profiling of different spatial locations, in this particular case, in the cerebellum of mice, from control and disease mice, in the context of SCA1 disorder. And we are able to show that the organization of the Purkinje cells, which are the specifically the cells that are known to be the most affected in SCA1, is completely losing its spatial organization if you look at the deconvolved cell proportion. But those changes are not really that visible when you look at simply the PP1R17 gene marker, the canonical gene marker for these Purkinje cells. And in fact, if you look at the change in this gene expression between SCA1 and control mice, it is not significant. But if you look at the change in cell type proportion using our deconvolved bulk data, mini bulk data, we're finding a dramatic loss of Purkinje cells in SCA1. And you can see here our deconvolved cell profiles in black, which are learned without any reference directly from the mini bulk experiments. We've also developed a deep learning method for augmenting single cell data with spatial information by training a deep learning model to predict a lower dimensional manifold within which we can then project the XYZ coordinates and see that, in fact, the model is, in fact, learning spatial information under the constraint in training that cells should be in this latent space, similar in distance to each other as they are in the original training data. And what we're finding here is that we're capturing a lot of these neuronal layers, both for neurons, but surprisingly also for glia, that oligodendrocytes and astrocytes are in fact showing a very similar layer organization from their single cell data sets, enabling us to now study the coordinated changes that are happening between glia and the neurons that they surround. We've also analyzed different subtypes of microglia, taking 300,000, or sorry, just let me see the slide. So 150,000 single cells that are showing signatures of microglia, and then distinguishing inflammatory versus synaptic microglia. And what this allows us to do is, in fact, show that Alzheimer's disease is very associated with inflammatory microglia, whereas schizophrenia is very associated with synaptic microglia instead, both in the differentially expressed genes, but also in the GWAS enrichments. Just a few weeks ago, we posted a paper dissecting the human vasculature and the BBB in the brain of both control and HD individuals by doing in silico sorting of 24,000 nucleotides, but also human ex vivo from epileptic surgeries to enrich specifically for vascular coupled cells across 90,000 nuclei, of which 7,000 are vasculature coupled. And we're finding that one of the subtypes of fibroblasts is specific to humans. We're finding zonation differences in pericytes and smooth muscle cells and endothelial cells with very different marker genes across all of them. And we're also finding that vascular cells play a very big role under HD and also AD in work that we have not yet published. We've used the single cell RNA data in red conjunctly with single cell attack data of DNA accessibility in green, projecting them both in the same subspace, and then using that to align individual cells for DNA accessibility and for RNA expression jointly in a coordinated fashion. We've used that to look for coordinated differences between control and disease individuals in the case of transcription and DNA accessibility, where these genes here are changing in coordinated fashion, both in their expression and in their DNA accessibility across all of the major brain cell types. And you can see here examples of dozens of genes changing in a coordinated fashion, with 75% of the changes being consistent and only a minority of changes being inconsistent between the RNA modality on the x-axis and the DNA accessibility on the y-axis. We've used the single cell attack data to also localize genetic variants associated with these diverse psychiatric and immune disorders, with the realization that psychiatric disorders are very much associated with excitatory and inhibitory cells, whereas Alzheimer's, just like many immune disorders, is in fact instead implicating microglia. And we've also looked at the upstream regulators of those genetic variants and predicted a number of cell type specific regulators that are changing in Alzheimer's disease, as well as predicting causal variants that will localize very specifically in disease-enriched cell types. Lastly, we've looked at the changes that are happening at the transcriptional level and what they're telling us about changes that are happening at the mutational level by looking at whole transcriptome SMART-seq data across 5,000 cells from 37 individuals, and then finding individual nucleotides that are mutated as inferred from the whole transcriptome sequencing, and how these mutations are in fact correlating with different post-mitotic or pre-mitotic cells, different levels of amyloid, different sexes, and also that the genes that are showing this excess in somatic burden are in fact very different between excitatory neurons, oligodendrocytes, and other cell types, suggesting that this additional somatic mutation burden is in fact influencing in a causal way Alzheimer's disease. We've integrated single cell data and bulk data with GWAS and QTLs using deconvolution across 3,000 individuals in a paper that we posted just a few weeks ago in bioRxiv. And we're finding these coordinated changes in cell type proportion across individuals with increased excitatory neurons associated with increased cognition and decreased associated with amyloid. And we're finding how these cell type proportions are changing between different sexes and between different ages, as well as predicting candidate driver genes that are associated with these cell type proportion changes. We're also doing integration across multiple subsignatures of Alzheimer's, looking at neuritic plaques and amyloid beta, neurofibrillary tangles and tau, as well as neuroinflammation, and how the locations in the genomes that are epigenomically associated with different subsets of these phenotypes are in fact implicating very different processes and very different pathways. We've expanded that to the electronic health records of millions of individuals by building hierarchical models that allow us to learn jointly across many different data modalities. And we've integrated these resulting phenotypic classifications of patients with genetics of these patients and with predicted expression downstream of these genetic variants to localize specific subgroups into specific pathways and genes that are changing in expression. Then lastly, we've been looking at how changes in exercise and diet are affecting multiple tissues jointly, looking at both human and mouse, and looking at single cell changes that are happening across small intestine, skeletal muscle, visceral fat, subcutaneous fat, and the pathways that are implicated. And in particular, we're finding that the effect of exercise is in fact combating the effect of nutrition and high-fat diet, and implicating both adipose stem cells as well as immune cells and T cells, suggesting again an interplay between these early adipocyte differentiation that was also the basis in our FTO work, looking at the decision to become a fat burning or a fat storing cell, and also the interaction of that with immune cells and resident immune cells in those tissues. We've used a multi-tissue approach to start relating peripheral blood with brain changes in AD versus control individuals. And we're finding that macrophages in particular in the circulating blood are very strongly associated with prediction for Alzheimer's disease. And we can see here that we can actually have a very strong AUC curve in distinguishing between risk and non-risk individuals in the context of simply their blood epigenome profiling. We've used the differences between chronological age and biological age inferred in our samples to define a delta age variable of the rate of aging for different individuals, and shown that delta age and ApoE4 are in fact independent and synergistic contributors to Alzheimer's phenotypes. In the context of regular normal aging individuals, we don't see a very strong effect of ApoE4, but in gray here, but in the context of these fast agers, we see a dramatic increase in the impact of ApoE4. And the pathways associated with delta age and ApoE4 are in fact quite different from each other, suggesting indeed that they are different contributors to Alzheimer's. We just published a paper looking at the progression of cancer across different stages of treatment and the coordinated changes that are happening in a longitudinal fashion in genetic, transcriptional, and epigenomic progression of disease, and the role of the immune microenvironment in these changes. And then lastly, we developed many methods for systematically testing our dozens of candidates in the context of imaging data with Kevin Egan and Kevin Smith in his lab and Maria Cusi in my lab, looking at synaptic density and how the synaptics changes across time and different conditions. And we've also developed these modular and programmable CRISPR-Cas9 and dCas9 constructs that allow us to modulate both the expression of a gene through inhibition or activation of their enhancers, through single nucleotide editing, and through knockout of both promoters and genes in iPSCs, which are then differentiated into different brain cell types. And we've also developed methods for doing these kind of perturbations in 384 well plates, enabling us to test many more candidates. And we've also developed methods for testing 7 million enhancers in a single experiment and inferring high resolution maps of where are these regions coming from in terms of their activity, localizing them into individual nucleotides, which allows to both predict their upstream regulators and also start understanding their impact in disease variants. Lastly, in collaboration with Andreas Penning, we're testing tens of thousands of these genetic variants in the brains of mice in different cell types, enabling us to now study how these are performing not just in vitro, but in the context of a living animal. So what I told you about is reference epigenomes to predict disease-relevant tissues and cell types and regulators, how we combine genetic, epigenetic, and transcriptional variation in disease, how we dissect disease at single-cell resolution across epigenomic and transcriptional profiling, and how we can discover cell-type-specific genetic effects by integrating single-cell and bulk data, and how we can expand that to multiple phenotypes, multiple tissues, and high-throughput validation. This is beautiful collaboration between the Kellis lab and the Tsai lab in the context of Alzheimer's and multiple other labs in the Boston area. These are some of the folks who are leading the work, and many of them have in fact gone off to start their own independent faculty positions. So we're always looking for talented postdocs. So please let me know if you're looking for positions, either on the experimental side or on the computational side. So I'll stop there with my acknowledgements and be happy to take any questions. Thank you.
Hey, great talk. Thank you very much for this exciting data. So maybe I can start with the first question. So finally, one of your major focus is the brain. You know, so you have done a lot of Alzheimer's. You have done also blood vessels. You have done psychiatric disease, so and so forth. And so I would like to ask you, did you work also a little bit on brain tumors? Have you looked on the microglia in the brain tumors? Because in the Alzheimer's disease, you clearly showed that the microglia is an important component. So I'd like to know, is the microglia also in your hands an important component in brain tumors? So in
we haven't looked at glioblastoma or neuroblastoma, and this is an area that we're super interested in, but it's all dependent on specific collaborations, specific partners, and availability of samples, etc. So if you guys are interested in such collaborations, this is definitely an area that I would be very interested in.
Yeah, we are extremely interested in that. So
great, let's follow up.
So we have projects on glioblastoma here.
Great.
So that's great. Okay, thank you very much. Then the other question that I have, and afterwards I will read questions that come up here. The other question I have is the difference between men and women, because it's extremely interesting that you find clearly the difference in, I mean, in a number of disease states. And also when I come back to the brain tumor stuff, it has been described that the transcriptomic profile in glioblastoma between men and women is different. So maybe this is a general theme that
I completely agree. I think this is something that we're going to see over and over again, where we basically are able to, you know, systematically understand the differences in the transcriptome, in the epigenome, and also in the disease response between men and women. And pretty much everywhere we have looked, we have found dramatic differences. And these are sometimes cell independent based on the sex chromosomes and their expression and all of the downstream genes that they control. Sometimes they are hormone dependent, where there's an interaction between the sort of quote unquote same genetic circuitry, but in the context of different hormonal stimuli, the response is in fact different. So this is, I think, going to be an area of major research across our field, something that we are still at the infancy of.
Yes, so okay. And the other one question, and then I will read, is in fact with the aging. So you have compared the fast agers, those that are aging fast, to normal ones. But did you also examine a group with slow aging people that are very young, even at let's say an old age?
Yeah. So absolutely, and the results are the converse exactly. So basically, the reason why we're I only mentioned half of the equation because the results are in fact defined in relation with the other half. And if I show you the, I'm going to share my screen again. There you go. So basically, here you have the slow agers, the fast agers, and the quote unquote normal agers. And what you can see is the effect of ApoE4 based on having 1 or 2 alleles of ApoE4 versus 0 alleles of ApoE4. And what you can see is that only in the fast agers is ApoE4 having this dramatic effect. Whereas in the slow agers, even if they carry a copy of ApoE4, they have normals for Alzheimer's. And then the slow agers have reduced risk of Alzheimer's, whether or not they have ApoE4. So this is basically telling us that both the fast agers have this synergistic effect and the slow agers have a protective effect. And even though it's not significant, you can see here a nominal difference between the E4 allele versus the E2-3 alleles in those individuals. So yeah, absolutely.
Yeah, that's very interesting. So now I'm reading questions. So the first one is, I was wondering which deep learning approach do you develop to predict spatial positioning in the context of single cell augmentation?
Oh, that's a great question. So this is work that's led by Juna, Na, Guillaume, and Fatima. It started both in my lab and in the context of a class project for a class I was teaching last fall. And what these students developed is a fully connected neural network, a multi-layer neural network that allows them to predict the spatial positioning of these cells using a data set of developed by Xiao Wang, who now has her own independent lab at MIT and at the Broad Institute. That's basically looking at the context of spatial positioning of cells profiled with 1,000 genes over multiple cycles of this machine. And what we're doing is asking, can we predict these XY coordinates, not directly as explicitly predicted, but instead by asking whether in the latent space projection, pairs of cells are constrained to have the same pairwise distances as they had in the original space. So what the method is learning is not an explicit three-dimensional position, because this is not super interesting because it's extremely experiment dependent, but instead to learn some latent space under the constraint that the distances match. And what's really remarkable is that the resulting analysis is in fact capturing a lot of the spatial information where the first dimensional variation of our data is in fact indeed capturing the cortical layers and additional residual information for these cells, enabling us to now start placing both excitatory neurons across different layers, but also you can see here the distributions of these glial cell types. So I hope that answers your question, Masha.
Yeah, I think it does. Yeah, many thanks, Manolis. So as far as if I understand correctly, basically you're minimizing the your loss will be trying to minimize the distance in the latent space, correct?
To minimize the difference in distance.
Yeah, the difference in distance. Yeah, sorry for the imprecision. Yeah,
between the latent space and the original space. Correct.
Okay. Cool. Yeah. Cool. Thank you.
Thank you.
Okay, great. Thank you very much. So maybe another question. So, I mean, you did all the studies on the vasculature. So I would like to know on the vasculature, so how it is comparing, how do you compare your data to the data that have been published in mice? Because, you know, you have a lot of data with a number of teams that have been done, I mean, single cell transcriptomic of the vasculature in mice in the brain. So what is your so how do you
yeah, that's also a great question. So what we did is that we actually co-clustered the human cells, both from the ex vivo samples of living humans and the post-mortem frozen samples and the mouse. And what we can see here is how are they capturing different subsets of endothelial, ependymal, oligodendrocytes, neurons, astrocytes, fibroblasts, smooth muscle cells, and then pericytes. And what you can see here is that indeed the human and the human frozen are in fact very strongly agreeing with each other. And then the mouse is capturing some aspects of neurons, astrocytes, and oligodendrocytes that are simply absent from the human frozen because we specifically in silico sorted against these cell types. But overall, there's a strong agreement, except for, of course, subtle subcell type changes that are found. In particular, the mouse is missing this whole subclass of fibroblasts that is only found in the human, which we believe is in fact a human-specific cell state of fibroblasts. And then also in the vasculature, we're finding this dramatic zonation, not only for endothelial cells, but also for pericytes and smooth muscle cells, where we can basically distinguish the venial versus the arterial and the specific gene markers of zonation, as well as these additional zonations that are happening between smooth muscle cells and pericytes with almost two orthogonal axes of variation, and some cells that are actually quite different between the two.
Yes, right. And do you see also difference between the tip cells and the stalk cells, because you have the tip cells on the edge of the vessel?
So we haven't specifically looked at those, but I would love to interact. If you have specific marker genes, we can send you information for the tip cells specifically.
Okay, thank you very much. So that's interesting. So I don't know. So are there any other questions in the audience here? Because I don't see any other questions anymore on the chat. Here.
Again, if you don't want to type up your question, feel free to just unmute and ask your question.
Yeah, please unmute. I mean, so go ahead. So maybe so you work on cancer. So let's talk about your work on cancer. So which tumors you did analyze? You did analyze. So
we've been focusing on metastatic melanoma right now in a collaboration with Geneviève Lalonde specifically, and we also published a paper in bioRxiv on prostate cancer, looking at the convergence of multiple different pathways in sort of target genes and diverse enhancers connecting together there. But melanoma has this very interesting biology where immunotherapy has been extremely successful, but for the subset of individuals for whom it works. And for about 50% of individuals, it just doesn't work. So what we're trying to do is understand what does the epigenome and the transcriptome of those cells and their microenvironment tell us about the, number one, what are the changes that are happening during disease progression? Number two, what are the changes that are happening in responders versus non-responders? And number three, what can we tell about predicting future responders and future non-responders from their pre-treatment differences? And because we have longitudinal data of single cell dissection of tumors before and after treatment, we can ask what are the predictive features beforehand that allow us to predict metastasis. So this is a paper that we submitted a few weeks ago, and I believe we are posting it on bioRxiv pretty soon. So we'll be.
So you have it in bioRxiv. Good. Because the interesting point is, if you have brain mets, for instance, the brain mets are a little bit specific. So since you are very heavily interested in the brain, so I think it would be nice to look at the brain mets, I mean, in the melanoma, because I mean, there should be specific signatures, I mean, in the brain mets going on when you compare to other sites.
I completely agree. These are fantastic questions and super interesting topics.
Okay. So are the students there or not? So where are the students? Students, you are still there? Students, if you have don't be shy, so you can ask.
So I see Ocean is turning on his videos.
Okay.
It's Lornella, and I'm using Ocean's computer, but I don't know, the video isn't working.
No problem.
So great talk. Great talk. Thank you very much for your talk. I was wondering how do you manage to do all these bioinformatical work? It's impressive.
I have an amazing team, amazing collaborators, wonderful students, and it's really just a very addictive work. So I'm up at 4 a.m. every day until 7 p.m. and I am living the life, working so closely with my entire team and with our collaborators. It's, I think many people would say that it's a little crazy to work on so many different topics, but I would say that they are self-reinforcing, that basically by working on so many different areas, we have idea cross-fertilization across them that I think is extremely beneficial to every one of those projects. And the way that I think about this is that it's a little silly to be studying glioblastoma in isolation without understanding the diversity of immune cells circulating across the body, the diversity of gene regulatory functions, the diversity, like basically for almost every disorder that we need to study, we are realizing more and more that these disorders are extremely polyfactorial, that basically there's just every single tissue is communicating with so many different other tissues, and every single pathway is implicated in many, many different disorders at the same time. So I think the ability to study disease from an integrative systems level global approach is very much the way of the future. And by forcing ourselves to work not as generalists, but as multi-specialists in so many different disorders, we're basically working with the best in the field for every one of those disorders, and also bringing in a perspective that many people who are focused on only one field don't have. And that's why I think that this cross-fertilization is really a benefit rather than a distraction.
Yes, thanks so much. It was so inspiring to see so much work, and yet I agree that it's great to have a global idea of everything. So thank you very much.
So,
and,
yes.
Wonderful name, by the way, for a Greek person to watch a name as Ocean is really awesome. Your parents are very creative.
So so one thing is that you are very highly active, and even you wanted to give your talk at 5 a.m. You told me you wanted to give your talk at 5 a.m. because I told you it's in general 11. I said, no problem. I can give my talk at 5 a.m. Okay, so that's you are extremely that's extremely active and so a little bit workaholic, let's say.
It's I mean, I will put it differently. I found people to pay me for my hobby, which is science. So I just
yeah,
do my
right.
hobby.
That's yeah, sure. Okay, so that's great. Okay, so I think we have so we have done it for today. So I would like to thank you very much, Manolis, for your talk. I mean, it's very impressive data. So you will be here normally in August, right? So we'll be.
My wife grew up in the Bordeaux region, so we have lots of little cousins that our children love spending time with. Okay. My wife is number 2 of 6 children, and every one of those children has 3 children of their own. So it's just a marvelous family reunion every summer.
So when you will be here, what? So
probably the first 2 weeks of August. We're spending most of our time in Greece. And then we're hoping to be in South France, or at least Atlantic France, for the first 2 weeks of August.
Okay, so I'm there, so we can, so we keep in touch.
Yeah, we'll drive.
We will meet for a good Bordeaux wine and something like that.
Sounds like a plan.
So thank you very much for everything. I will send you. We will ship you the wine also before. Okay.
My wife and I will love it. Merci beaucoup, Andreas, and thank you to everyone.
Okay,
bye.
À bientôt, hein. Salut. Bye. Salut. Ciao.